Our next discussion will be about gametogenesis, including spermatogenesis and oogenesis. So gametogenesis is the process of forming the gametes, and it is the process whereby a haploid cell is formed from a diploid cell through meiosis and cell differentiation. Spermatogenesis is the formation of the sperm, while oogenesis is the formation of the egg. So the reproduction in vertebrates is actually by sexual means. So it actually involves haploid germ cells, which includes your ovum or the female component, and the spermatozoa, which is the male component. So both arise through meiosis, which is the cell division where each daughter cell receives half of the genetic material from the original cell. So primordial germ cells is derived from the extraembryonic endoderm or the yolk sac, which then migrates to the gonads. So stages in gametogenesis includes the following. First, pairing and doubling of the chromosomes in oogonia, followed by the growth as primary oocyte. And then next would be the first meiotic division that produces twice of the cells or the secondary oocytes. And then second meiotic division produces 4 haploid cells, which includes the spermatids and the ova. And then the spermatids mature and differentiate to form functional spermatozoa. So in spermatogenesis, all 4 sperm cells produced are viable. In oogenesis, only one of the 4 cells produced is viable. Other become abortive as polar bodies, and that later degenerate. For the sperm formation, the sperm are produced in long-coiled seminiferous tubules in the testes. Spaces between the tubules actually contain the testosterone-secreting cells, which feed on the sperm cells. Okay, so diploid male germ cells or the spermatogonia line the tubules in the inner wall, and then the spermatogonia divides repeatedly by mitosis and differentiate to become primary spermatocytes. Meiosis of primary spermatocytes produces immature sperm or the spermatids, and we have the Sertoli cells inside the tubules to support the developing sperm and help regulate sperm production. So in here, you will see how it looks like for the cross-section of the seminiferous tubules inside a testis. Again, this is the membrane, outer membrane, and then adherent to it would be the spermatogonium, which will undergo mitosis to become primary spermatocyte. And then after meiosis 1, it will become secondary spermatocyte, and then after meiosis 2 would be the immature sperm cell, and then eventually leading to the formation of the mature sperm. So then again, as what have been discussed, Sertoli cells do actually help when it comes to the nourishment of this or the nourishment for the cells to be developed. So during spermatogenesis, 4 sperm result from each primary spermatocyte. So the process also maps onto the physical structure of the wall of the seminiferous tubule with the spermatogonia on the outer side of the tubule and the sperm with their developing tails extended into the lumen of the tubule. So again, from the spermatogonium, it will undergo mitosis to produce primary spermatocytes. These primary spermatocytes will then be divided into two, okay, or two cells will be produced during the secondary or during meiosis 1, and then after meiosis 2 it will now give you 4 identical spermatids, and then it will definitely differentiate to 4 mature sperm cells. So for the sperm structure, the spermatozoa from different animals have a wide variety of forms, but all have head and tail regions. So the head region serves with 2 functions. The first one is to contain the nucleus or the genetic function. So it does have an acrosomal cap that contains enzymes that allow sperm to break down membranes around egg and fertilize it. So the tail is what you call the flagellum that provides the motility, and then the meat piece between the head and the tail contains mitochondria that provide ATP to fuel swimming. Okay, so it's very important that the sperm structure does have a lot of, the ATP for energy, so that it has the ability to swim going to the egg. Okay, because as you know, when the sperm travels going to the egg, it's like also a survival event. Okay, so some of the sperm would actually die in between the course, but then again, there are also some sperms that could actually reach the egg, but then again, it also boils down to which sperm could actually penetrate the egg itself. Okay, so the regulation of the sperm production. So gonadotropin releasing hormone is secreted by the hypothalamus that stimulates the anterior pituitary to secrete luteinizing hormone and follicle stimulating hormones. So then again, with this hormones, they are the ones who are, regulating the formation and development of the sperm. Okay, so the luteinizing hormone binds to the testosterone secreting cells, causing them to increase their output, and testosterone and follicle stimulating hormone binds to the Sertoli cells and encourage sperm sustaining activity. So in here you will see how the sperm looks like from the beginning up to the end. So in here, you will see that hormones control sperm production in a negative feedback system. So when you say a negative feedback system, it allows a positive, or it does have a positive and a negative um, effect, okay, on the cycle or on the loop of the process. Okay, so here you will see positive or triggering factors, and then once these triggering factors already affected the testes, there will be an inhibition or inhibiting factors that would actually control the hypothalamus and the anterior pituitary. Okay, so pituitary hormone effect. So the pituitary hormones um, affects the following. So we have the luteinizing hormone and the follicle stimulating hormone from the pituitary gland that stimulates spermatogenesis and testosterone secretion by the testes. So hypothalamus, you have the gonadotropin releasing hormone. So once this gonadotropin releasing hormone affects your anterior pituitary, it releases now your follicle stimulating hormone and the luteinizing hormone. So what does this hormones affect? We have your testes. So in the testes, the FSH affects the Sertoli cells to facilitate spermatogenesis, and the LH2 um, help with the Leydig cells. Okay, so when in reaction to these hormones, of course, the Sertoli cells will actually produce inhibin affecting the anterior pituitary. For the Leydig cells, it can affect the Sertoli cells, or it can also secrete the testosterone inhibiting anterior pituitary and your hypothalamus. So the testis hormone effects, so testosterone and inhibin inhibit the secretion of GnRH by the hypothalamus and LH and FSH by the pituitary. Okay, so that's how um, the cycle of the sperm production is being regulated by your different hormones. So the route sperm travels. So the sperm is produced by the Sertoli cells in the testes, then released into the lumen of the seminiferous tubules, and then sperm travels to the retitestes and enter the efferent ductules. This is actually the point where the sperm exits the testes, and then the sperm then enters the epididymis. This is the site where the sperm mature and stores the matured sperm then make their way to the vas deferens that connects to the seminal vesicle, joining to form the ejaculatory duct. This is the point where sperm is mixed with fluids from the seminal vesicles. So this is also the duct that penetrates through the prostate gland to join the urethra. So this is where both urine and sperm can pass through, but of course it cannot be that it cannot accommodate both urine and sperm at the same time. So there should be, it can only be sperm for a time, and then urine for another time. Okay, because if urine um mixes with your sperm, of course, definitely the sperm will die because of the acidity that is found in your urine. Okay, so then the sperm then flows and receives more substances from the bulbourethral glands and eventually to the penis, or the male organ of intercourse. So that's how the sperm travels from the um, testis going out of the penis. So for the oocyte maturation, female germ cells do not divide after birth. So I have already discussed that with you. A girl is born with about 2 million primary oocytes, or immature eggs, suspended in the prophase 1 of meiosis in her ovaries. At puberty, hormonal changes prompt primary oocytes to mature one at a time in an ovarian cycle about 28 days. For the egg membranes and structure, cytoplasm enclosed with plasma membrane is found. You will also see the vitelline membrane, which is a thin membrane closely attached to the plasma membrane. And then the zona pellucida is actually a glycoprotein layer that's found in mammals. And then you also have the corona radiata in mammals. This is a layer of follicle cells that becomes sloughed off after fertilization. So this is how it looks like. So in a schematic diagram, you will see how it looks like when the egg is being penetrated by the sperm. So you see here acrosome reaction. So in here, you will see the um, different parts of the egg. You have cortical granules, you have the vitelline membrane here, pre-vitelline space, the zona pellucida outside, and then the corona radiata. So all of these layers of membrane enclosing your egg cell will actually be protruded by the head of, or the acrosome of the sperm. Okay. So under the microscope, it looks like this. So this is the zona granulosa. This is the antrum, or the space in between. This is the oocyte itself, the zona pellucida, and then the corona radiata that's um, surrounded by it. Okay. So the process of oogenesis occurs in the ovary's outermost layer. So before birth, you have your the oogonium and that undergoes mitosis and then produces a primary oocyte and being arrested in prophase 1. So after puberty, meiosis continues. You will be producing a secondary oocyte that arrests in metaphase 2 and then also produces the polar body. And then during ovulation and sperm entry, you will produce here um, an oocyte, and then meiosis and fertilization, you will be having a fertilized egg and another polar body. So steps in the ovarian cycle. So first would be an ovarian follicle begins to mature, and then the primary oocyte enlarges and secretes proteins, and cells around it divide. Second, a fluid-filled cavity begins to form in the follicle's cell layer, and then the primary oocyte completes meiosis 1 and undergoes an equal cytoplasmic division, forming a large secondary oocyte in the first polar body. Next, the ovary wall ruptures during ovulation. So the secondary oocyte enters an adjacent oviduct, and then the cell cycle is halted in metaphase 2 and will not continue until fertilization. So as you see, there's always this, two stops. So the first one is before puberty, which is the arrest in prophase 1. And then not until the egg is being fertilized, it will also be arrested in metaphase 2. Okay, so follicle cells left behind after ovulation develop into a corpus luteum that secretes hormones. And if pregnancy does not occur, the corpus luteum degenerates, and eventually females would have what you call the menstrual period. Okay, that's to release the unused egg out of the body. Okay, so this is how the corpus luteum look like. So you have here the theca. Okay. So once it's actually degenerated, it would look like this, a corpus albicans. Fully degenerated corpus luteum. So this is how the ovulation process looks like. So again, from the ovarian follicle, you have here the development of the follicular cells going to the primary oocyte, and then here you will be producing the first polar body and the secondary oocyte. And then if there's ovulation, it looks like this. And then of course, it will eventually become a corpus luteum. Okay, so that's how the schematic diagram of the um, oogenesis looks like. So the regulation of the egg production. So in here, we have two, okay, two regulation of the egg production. So the first one would be the follicular phase, wherein the hypothalamus secretes the gonadotropin releasing hormone, affecting the anterior pituitary. So the anterior pituitary will then release FSH and then LH, affecting the ovaries. Okay, so for the reaction of the follicles, it will then release estrogen, okay, to um, affect the uterus endometrium, or it can also affect the anterior pituitary and then the hypothalamus to stop the generation of the GnRH and FSH and LH. So again, the pituitary hormone effect is that LH and FSH stimulate several follicles to grow. So as long as the pituitary, anterior pituitary releases FSH and LH, of course, it will definitely push on the growth of the follicles. Okay? So ovarian hormone effects, follicles produce low levels of estrogen that would eventually inhibit GnRH secretion by the hypothalamus, keeping LH and FSH in levels that are low, and cause endometrial arteries to constrict due to low levels, resulting in menstruation. Okay? And then the other one would be during ovulation. So in here, you will see that the hypothalamus produces GnRH, affects the anterior pituitary. It will release FSH and LH, affects the ovaries and follicles. So with the estrogen being released by the ovaries, it will affect your uterus, especially the endometrium, and the estrogen also, um, affects your hypothalamus. So if, as you see here, for the ovulation process, it looks like a positive feedback loop compared to this one that has a positive and a negative loop. Okay, so in here it really just promotes ovulation. So the more that the hypothalamus is being um, affected or being um, or gets the right amount of estrogen, it will also increase its yield of the gonadotropin releasing hormone, affecting the anterior pituitary, and of course the amount of the pituitary hormones affecting the follicles. Okay, so yeah, that's how the regulation of egg production happens. So again, remember that it has two phases, for the follicular phase and then for the ovulation phase. So next, we also have the luteal phase. So the ovarian and menstrual cycles of female reproduction are regulated by hormones produced by the hypothalamus, pituitary, and the ovaries. So in here in the luteal phase, again, you have the hypothalamus secreting GnRH, affecting the anterior pituitary to release FSH and LH, affecting the ovaries and the corpus luteum. Okay, so it has a negative feedback, the estrogen and the progesterone to the uterus. Also, the same hormones do have negative effects as well in the anterior pituitary and the hypothalamus. Okay. So with the ovarian hormone effects, the the effects would be blockage of the GnRH production by the hypothalamus and LH and FSH production by the pituitary, and this causes the endometrium to further develop. Okay. So this is the comparison of how the egg and the sperm formation looks like. So in here, you again, from what we have or what I have explained earlier, for the egg formation, only one egg will be viable, and then the other three of the um of the produced cells would be polar bodies, and then for the sperm cell, all of the four developed cells can be viable for fertilization. Okay. So these are just images for you to check about spermatogenesis, how they look like. Okay, in arrangement of the basal lamina, how does the spermatogonia and spermatid look like? Okay. And this is for oogenesis. Okay. So this is how the different polar bodies are being produced and how a follicle looks like. Okay. So again, to summarize the key concepts of gametogenesis. So oogenesis occur in the ovary with a follicle of epithelial cells. On the other hand, spermatogenesis occurs in germinal epithelium lining of the seminiferous tubules of the testes. And oogenesis begins with the oogonium, and spermatogenesis begins with the spermatogonium. So both are normal 2N cells or haploid cells. And reduction in chromosome number is accomplished via two meiotic divisions. Okay? So that ends our second lecture for today. If you have any questions, just let me know and I'll be answering them. Thank you very much.